we're very pleased to welcome Roger Barker, who is lecturer in neurology in the medical faculty here in Cambridge. I'm sure known to quite a few of us here. And he's based at the Cambridge Centre for Brain Repair in the Department of Neurology. And Dr. Barker has been very much involved in cell-based therapies, including xenografts, as well as stem cell therapies for quite a wide range of brain diseases. So he's very, very experienced in this field, and I think is an ideal speaker to come and give us our talk today on stem cells. It's entitled Ethical and Theological Factors in the Use of Stem Cells for Brain Repair. So thank you very much, Roger.
Thank you very much for inviting me. What I thought I would do in this talk, I'll get around here, is I thought I would basically just take you through a little bit on the background. And by background I mean to talk a little bit about Parkinson's disease and how people see cell therapies working in neurological disorders. But essentially I'm going to be concentrating on Parkinson's disease, the use of cells to repair the brain in Parkinson's disease, explain where the field has got to, and then sort of move on to stem cells, what we mean by stem cells, and then run through what I see to be the major ethical and theological issues with respect to that. I have to say this is a very personal view. This is my interpretation of scripture. This is my interpretation of the argument. This is very much my position as I see myself positioned within this field, which is quite a difficult one of where one should stand with respect to the use of stem cells in medical science. And so what I'm going to start off with is just to, first of all, illustrate that this is not a topic which is not in the public eye, if you like. So this is the Guardian from 2001, and this was a headline which referred to a transplant trial which took place in Parkinson's disease, which produced negative results. And we do a transplant trial, or are involved with a transplant trial in patients with Huntington's disease, supported by the Medical Research Council, and that headline caused MRC head office to go into a major nosedive, and I had countless phone calls that day and had to convene an urgent meeting to discuss the future of our cell therapy trials in Huntington's disease. And this is, I think, a very important issue, which is the public perception on the use of cell therapies, which are very exciting. And as I've said before to some of you here, the problem with the press is that there are only two outcomes with any new innovation with respect to medical science. They're either miraculous or they're catastrophic. And this is the problem you have to deal with, is that there is hyperbole that is used in these issues, which make it sometimes very hard to actually dissect where we are with respect to some of these new innovations. In The Guardian last week, you can spot which paper I read, on Thursday there were two articles relating to Michael J. Fox and his advert which had appeared on American television supporting a Democratic nomination, where he was accused by an individual of manipulating his therapy for his Parkinson's disease to make his condition look worse. He was arguing for the use of embryonic stem cell research to be used in America, or to be federally funded to help with Parkinson's disease, which is the condition that he suffers from. And on the following day, on Friday, there was an 8-page article by Zoe Williams entitled Abortion is Not Evil, where she was making the point that we regard abortion as a taboo subject, and haven't we moved beyond that, and we can just accept it as one of those things that happen, and actually we needn't be quite so sensitive about it. So issues to do with stem cells, issues to do with transplant trials in patients, issues to do with abortion are all very much at the forefront of the public's mind and are very much in the newspapers. So if we go back to consider how can we use cells in neurological disorders. Now I'm afraid this is a bit technical, but essentially I just want to make a few points. The first is that we can think of using cell therapies for the nervous system in many different ways for many different conditions, so that the implications of where we can go with cell therapies in neurological disorder is vast. So the simplest way to think of it is as ways of replacing cells which have died in disease processes. Parkinson's disease I'll talk about. In Huntington's disease, genetic disorder, we can replace cells. That's what we're doing a trial with at the moment. In Adam Brooks with human fetal tissue, you could replace the cells which are lost with stroke. In multiple sclerosis, you lose cells that insulate nerve cells in the brain. You could try replacing those. So you can use cells to repair the brain by replacing the things which are lost. Alternatively, you could use cells not to replace missing cells, but to deliver factors which make other cells in the brain grow more healthily. Sort of baby bio for nerve cells in the brain. So in case of Parkinson's disease, you might be able to deliver a factor which encourages the cells which normally die in Parkinson's disease to grow. You can think of them as ways of delivering missing chemicals, missing enzymes, so genetic disorders where people are born with abnormalities in certain pathways which prevent normal processes within cells. You could use the cell to deliver the missing enzyme. These are incurable and they often affect young children. You can think of them as ways of delivering drugs more effectively into the nervous system. So there's some evidence, for example, for brain tumours that if you've got stem cells, put in a drug therapy into a stem cell, it would migrate to the tumour, deliver the drug in high concentration and kill the cancer cells. And finally, you could think of them as actually just providing a bridge along which the natural repair processes in the brain, or the brain or the spine, could work. So I think the important point I'm trying to make here is that cell therapies have massive implications for neurological disease, and we tend to think of it in terms of Parkinson's disease. We talk about Alzheimer's disease. I think Parkinson's disease is the front runner, but actually cell therapies for neurological disorders has massive implications for vast numbers of conditions, most of which are incurable, many of which are fatal. But what I'm going to concentrate on, I just meant to make this point as well, with stem cells, there is also the possibility that you could use stem cells to actually model disease processes, growing stem cells out of patients who have diseases, or making artificial organs or artificial cell lines and using them to test drugs. And for example, on the news today is the team from Newcastle who have produced a small liver out of umbilical cord stem cells, which they feel will be a very good way of testing drugs rather than necessarily using them for liver transplants at this stage. So there are massive implications for the use of cells in the nervous system as well as for stem cells. But what I'm going to be concentrating on is Parkinson's disease and replacing missing cells for the first half of this talk. So what is Parkinson's disease? Well, I think most people know what Parkinson's disease is, because most people, I think, will have come across people who have it. The most famous person of recent times to have it was the late Pope, John Betjeman, the poet laureate, who's been celebrated his 100th birthday, I think this year, has had Parkinson's disease. So there are many famous people who have it, and in Cambridgeshire there is a new case of Parkinson's disease diagnosed every 5 days based on our own work, and a case which could be Parkinson's disease every 3 days. So 1 in 800 people get Parkinson's disease, 120,000 people in the United Kingdom have it. And the classic features of Parkinson's disease are the loss of a relatively discrete set of cells deep within the brain. So at the top of this thing here called the brainstem is a thing called the nigra. Nigra because it's black here, melanin in these cells, and they produce a chemical called dopamine. And it is the loss of those dopamine nerve cells and the formation of this pathological abnormality which leads to the clinical features, which are slowness of movement, a resting pill-rolling tremor, stiffness, and an unstable gait. So this is a common condition for which we have many therapeutic agents which can help the symptoms, none of which are curative. So the disease is ultimately fatal in the sense that we cannot arrest the disease process. We have no cures for it, all we have are symptomatic treatments. But nevertheless, knowing that it has this core pathology makes it an obvious target, or an obvious target for cell therapy, and that the rationale is fairly straightforward. So the rationale is, and you don't need to be Einstein to understand this, is you take a patient with a neurodegenerative disorder, which is, in this case is Parkinson's disease, you identify what the pathology is, which in this case is the loss of these dopamine cells in the bottom of the brain, in the nigra, so that is the key problem, and you simply say, I'm going to replace those and I'm going to transplant in dopamine cells, and if that is correct, then I'll cure the patient. So this is a very simple strategy, and the question is, how successful is this? Well, it's very successful. And I think this is very important because I think when one reads about stem cells, it's sometimes, and cell therapies for the brain, it's sometimes difficult, as I said at the beginning, to disentangle what's real and what's just hype. And this is a couple of patients who received not a stem cell transplant, but they received a transplant of human fetal dopamine cells. So these are 2 patients, and I just want to concentrate on patient 4. Patient 4 was a patient who in the late 1980s had had Parkinson's disease for about 10 to 12 years. As a result of that, he had developed major symptoms which were proving impossible to control with drug therapy. He then received, on one side of the brain, the transplant of 5 fetuses. And it wasn't the whole fetus, they took out the dopamine cells which are developing in the embryonic nigra. So in the developing fetus, you take out where the dopamine cells normally develop, you dissect that out, you collect that from 5 fetuses, you turn it into a cell suspension, and then you inject it into the brain. So this is the tissue from 5 8-week-old terminated fetuses, and it was transplanted into one side of the brain. The patient received drugs to stop the body rejecting it for 18 months. He's subsequently been off treatment for the last 15 years and was last reported in the medical literature 5 years ago, where he had a surviving transplant and was better at that stage than he was in 1988 when he received the transplant. In this slide, at the top, which you won't really be able to see, is his clinical progress. And the point here is, before he had his operation, he spent 2/3 of the day off. And that means that despite taking his drug therapy for his Parkinson's disease, he looked as though he wasn't taking any medication at all. So it looked as though he just had Parkinson's disease and the drugs were doing no good whatsoever. So 2/3 of the day he spent off with the drugs being ineffective. He then received this transplant on one side of the brain, and nothing much happened till about 3 to 6 months, when his off period halved. The miraculous thing, or remarkable thing about this chap, is that 3 years after he had that transplant on one side of his brain, he had been transformed from being off for 2/3 of the day to being on all day. So he was on all day as though his Parkinson's disease wasn't really there. That's a pretty remarkable improvement. It's even more remarkable when you think that happened in the face of all of his drug therapy being stopped. So 3 years after a transplant on one side of the brain, this chap had been gone from off 2/3 of the day, on all day, on no medication, which just never happens in Parkinson's disease. So clinically he improved, and when they did a scan which looked at dopamine, the thing that's missing in Parkinson's disease in the brain, and you followed him over time, at the site of the transplant you can see that there was no dopamine. It then increases at 1 year, and at 3 years you can see that there is a massive increase in dopamine at the site of the transplant. So you can clearly see that if you get it right, you can have a very dramatic effect on a patient. And as I say, 20 years down the line, this patient is still better than he was before he had his transplant. You can also see that those cells which have been transplanted have not been affected by the disease process. How do I know that? If you look at the patient's dopamine cells over here, or where dopamine is produced, you can see that whilst the transplant has grown, the patient's own dopamine system has disappeared. So this is a proof of principle. The transplantation of human fetal cells containing the developing dopamine cells, if transplanted into the right patient with Parkinson's disease, can have a dramatic effect on their condition, which you will not see with any other treatment. However, it's not as straightforward as that. That original headline referred to trials where things have not gone as smoothly as that. So there have been problems of reproducibility. There are also problems with using fetal tissue at a practical level. As I said, this patient required tissue from 5 fetuses in one side of his brain. If you wish to transplant a patient with Parkinson's disease on both sides, you will therefore need to simultaneously collect between 5 and 10 fetuses from a termination of pregnancy, identify the bit of brain that you're interested in, namely developing dopamine neurons, and transplant that in. So there are real practical problems. There are clearly issues of infection. Could I be putting in things like CJD-like material, other infections? There are immunological problems, but it's obviously the ethical dilemmas which I shall come on to discuss. But given these, there has always been a search for other sources of cells. And there have been many different types of cells which have been looked for, but by far the most attractive and by far the most represented in terms of publication and public perception are the use of stem cells. And it's very hard to pick up a medical journal these days without seeing something about stem cells. And why are stem cells so attractive? Well, the reason why stem cells are so attractive is that you can grow them in large quantities. So stem cells are essentially a cell that divides. They divide often into a daughter cell, which can carry on dividing, as well as a cell which can then differentiate into another type of cell. The point being, if I start with 1 fetus on day 1, by the end of the year I could have several laboratories full of stem cells. So the practical problems are circumvented within a very short period of time. So you can grow them to very large numbers, which gets around the delivery problem, Because they're dividing, they're relatively easy to manipulate. So if you wanted to put things into your cells which made them attractive, you could do that. And there are issues about their behaviour which may be attractive, namely their ability to be able to migrate to areas where there's damage in the nervous system, which may have great benefit. So they have the capacity of self-renewal, and they have the capacity to turn into different cell types. That's why stem cells are so attractive, is this ability of them to divide and differentiate. One of the problems, I think, with stem cells is that people think when you use the term stem cell, that just refers to one type of cell. There are many different types of stem cells which bring with them their own advantages and disadvantages. And I think it's very important to get that point across, because different stem cells bring with them different ethical problems. Most of the discussion will concentrate on embryonic stem cells, which are the cells which are derived from the very early embryo, just after it's been fertilised, and are derived from spare embryos in IVF programs. Those cells clearly have a very different behaviour and a very different ethical perspective compared to adult neural precursor cells, i.e. stem cells which are found in the adult brain. So every single one of us in this room has cells, new neurons being born in their brain all the time. So there are certain areas in your brain where new nerve cells are being born, so there are types of stem cells in the adult brain. They are clearly very different from those types of stem cells, but they will still be called stem cells nevertheless. There are clearly stem cells which you can get out of the embryonic brain, so these are brains from fetuses which have been collected through termination of pregnancies. They again have problems, advantages and disadvantages. There are stem cells which are taken outside of the brain and outside of the embryo in the adult, so taking cells out of the bone marrow. There are these things called bone marrow stromal cells which seem to have a capacity to turn into all sorts of different types of cells within the body. There are blood stem cells, and it's always important to remember when people say, do you think stem cells will ever enter into the clinic, to remember that stem cells in the blood world, hemopoietic, and the haematology world, have been used for years to treat people with cancers of the blood system. So there are blood stem cells, and then there are a host of other stem cells found at other sites. So there are many different types of stem cells. So if people say, well, I've got a stem cell that does this, the problem with my stem cell is that doesn't necessarily apply to all stem cells. One has to be quite particular about the different types of stem cells and what they, the advantages and disadvantages that they bring. In the sort of neurological condition, and with respect to Parkinson's disease, I think you can think of stem cells whichever source in one or two ways. You can either say, well, is there a stem cell in the brain which I could turn on? So the most attractive way to repair the brain, which would mean that I wouldn't have to give this talk at all, would be to say, well, there are stem cells in the brain that make dopamine cells. They don't work in Parkinson's disease. Why don't we just switch them on, cure Parkinson's disease, and that's that. And there's no ethics with that. There's no problem with that, because you're using cells which are there in the adult brain. There's no problems of delivery, because the patient's got their own cells. No problems of infection, no problems of immunology. Everything evaporates if we could just get the brain to switch on its own precursor cells, in this case to make dopamine cells, or, coming back to what I was talking about earlier, we transplant in exogenous stem cells, so things that we've grown in the lab. If we want to switch on the stem cells in the brain, the problem is we've got to find them, and at the moment they don't seem to exist. So one of the arguments, for example, against the use of embryonic stem cells or any other stem cell transplant is, why don't we just use the stem cells in the brain to repair the brain? Well, it begs the question, why aren't they doing it in the first place? Because if they did it in the first place, we wouldn't have neurological disease. But actually, trying to get new dopamine cells grown in the adult brain, so do the adult dopamine cells replace themselves? The answer is no, they don't. There doesn't appear to be any evidence for that. So the idea that you can actually fix Parkinson's disease by getting the patient to switch on their own dopamine stem cells seems not to be a reality at the present time, and I suspect those cells don't actually exist in the adult brain. So if we're going to transplant in the exogenous stem cells, then the question is, well, what are we going to use? And depending what we're going to use generates a number of ethical and theological issues, which I shall now run through. So I'm afraid some of these slides are a bit wordy, but I'm going to go through each of these points in turn. It would seem to me that the fundamental questions we have to address if we are to take stem cells exogenous stem cells into patients, the questions we have to debate are, if we think stem cells, cell therapy is a useful approach, why don't we use adult stem cells? And I've already talked a little bit about that, and I'll talk a little bit more about that in a minute. The second question and the second criticism, which I think is often levelled at the use of this work, is that the use of these type of cells is actually a bit of science fiction. It's not really going to impact on clinical medicine, and what people do is they put forward a very positive view on it in order to justify the use of these cells, when in reality they won't enter into the clinical domain. And I want to make that point, that I don't think that is, I think that's an unfair criticism based on what I've shown you already with patients, and I'll come on to say a little bit about what you can do with ES cells. Obviously, should we be using any other alternatives to cell therapies, and I'll briefly touch upon that. Then I think we get down to some really very difficult questions, and I think this is where one has to confess that there is very hard to know what the right answers are and where one stands in this about, as a physician, who it is that you are most responsible to. Are you most responsible to God, to the patient, to the scientific medical research? How do you square all of those, and how do you actually steer a course through that? I think a fundamental question is, when does the embryo acquire a soul? Because that clearly will impact on your views about when you're actually allowed to use stem cells as a cell therapy. And one of the questions which I think is very difficult is, even if you stand aside from this, at what point can you ever be seen to use therapies which have been derived in part from stem cells? And this has been a reality. There was great discussions about 10 or 15 years ago about a vaccine which was developed with the use of human fetal cell lines. The question was, it was mainly the Catholic Church, should they be doctors of a Catholic persuasion, should they be using that vaccine, given the fact that it was developed on human fetal cells, using human fetal cells? And that caused a great deal of angst and debate as to whether it was an appropriate thing to do. So I'm going to run through these points and then conclude by my sort of position on where I see myself sitting in this rather difficult and uncomfortable area. So the first question is, if cell therapies are useful, why are we wasting all our time looking at fetal and embryonic stem cells? Why don't we just use adult stem cells, which do not bring with them the ethical and theological issues which the other type of stem cells have associated with them? Now, I've already said that in the case of Parkinson's disease, I think the problem is that we don't make new dopamine cells from stem cells in the brain. So how you're going to repair Parkinson's disease by getting adult stem cells to work, at least in terms of those which are in the brain, seems not to be a reality. The other problem is that one of the problems with stem cells is, whilst you can get them to divide and to develop into all types of different cells, getting them to turn into dopamine cells, the cells you want for Parkinson's disease, seems to be incredibly difficult. So it is actually extremely hard to get a consistent number of dopamine cells generated out of any stem cells, and the one stem cell which does it better than any other are the embryonic stem cells, the ES stem cells. It's also important to remember that what you don't want your stem cell to do is, you don't want it to turn into anything else, and this could also be the problem with adult stem cells. They may actually turn into cells that you don't want them to turn into. There was a great deal of excitement 5 years ago when there were a large number of papers published showing that if you put adult stem cells into the brain, or transplanted them into animal models of disease, you could get them to turn into nerve cells. So this was done with blood stem cells, turning blood into brain, and it was also done with bone marrow cells. So in this, they injected adult blood and bone marrow stem cells into animal models, looked in the brain of those mice and rats, and found that some of those stem cells had turned into neurons, turned into nerve cells. And so this was a process which was called transdifferentiation. You take a cell which is destined to be something else, namely a blood cell, and you've managed to persuade it not to turn into a blood cell, but to turn into a nerve cell. Further analysis of those studies and further studies actually have shown that A, that is incredibly rare. So there was a great paper in Nature Medicine by Derek Van der Koy's group showing that he had done the blood to brain transplant experiment 247 times and had not found a single nerve cell. So it's a fantastic paper in Perseverance. It was also amazing in Nature Medicine to publish a paper that was entirely negative in every single respect. So it happens with very low frequency. The other problem was that when they looked closely, those cells which looked to be nerve cells had actually not turned into nerve cells, they'd fused. So the blood cell or the bone marrow cell had gone in, it had fused with a nerve cell, and it had turned into one big nerve cell, but actually there was no evidence that the cell had turned into a neuron. So we can't get sufficient numbers to turn into nerve cells. Those which do appear to be nerve cells are not truly nerve cells, they're just fusing. Even if we get them to turn into nerve cells, they don't look like the ones that we want them to. We have to get them to integrate in the brain, which nobody's been able to do because nobody's been able to do this. And the question, which comes back to something I said at the beginning, was if we have adult stem cells, adult brain cells, which are capable of dividing, why do we have neurological disease? If they're so good at fixing the brain, then they should fix the brain and we shouldn't have the disease in the first place. So are the adult stem cells going to be affected by the environment in which they find them? So whilst a lot of arguments are put forward for the use of adult stem cells, and it would be wrong for me to say that there is not worthwhile work being done in that field, at the moment, adult stem cells, for Parkinson's disease in particular, are not a reality. They are not likely to produce what we need them to produce, and thus there is nothing on the horizon in terms of these cells being useful. The second question is, is this all science fiction? Am I justifying the use of stem cells on unrealistic expectations and data? Well, I've already shown you that if you use human fetal dopamine cells, you can transform the life of a patient with Parkinson's disease. This paper from Nature 4 years ago showed that in Rob Mackay's lab in America, he was able to take mouse embryonic stem cells by exposing them to various factors in the laboratory, growing them up. He was able to turn mouse embryonic stem cells into dopamine cells, which he could transplant into an animal. They would survive, they would turn into the appropriate cells, they would reverse the behavioural deficit in that animal. If you looked at those cells in any respect, they looked like normal dopamine cells. So 4 years ago, you could get a mouse ES cell to look like a human, to look like a dopamine cell, which would be useful for Parkinson's disease. Human embryonic stem cells are fast catching up. So that human embryonic stem cells being manipulated and engineered to turn into dopamine cells is something which many people can now do. Whether they can do it consistently and with the fidelity which they've got with these mouse embryonic stem cells is, I think, still debatable. But nevertheless, you can make embryonic stem cells into dopamine cells. So this is not science fiction. This will be impacting on the clinic in the very near future, or at least people are going to have to resolve the issue as to whether we use these cells in the clinic. Should alternatives be used? I'm not really going to talk about this because it touches upon many different areas. Of course, there are many other therapies that people are thinking of with Parkinson's disease, better drug therapies, neurosurgery, making holes in the brain, repairing things, not repairing things, correcting circuits. Those are not curative. They're symptomatic treatments. There are the use of growth factors, and there are ways of actually trying to stop the pathology. So there are lots of other things that are being tried, but none of them, in terms of this, has just hit a major hurdle in America. So the trial which was using this to repair Parkinson's disease has now been halted. These have not really got into the clinic yet. They all bring with them problems. And there's no reason to believe that these therapies are not worthwhile pursuing. But at the moment, the most successful therapy clinically in terms of curing patients, if you will, of Parkinson's disease has been the use of human fetal dopamine cells. So that is, I think, trying to deal with some of the criticisms about how realistic are we about where we're going, and are we preferentially highlighting stem cells because it's to our advantage to do that whilst playing down other therapies. I think we now get to some more fundamental questions, which is, to whom is the physician most responsible? And I sit in the middle here, I would say. I am a Christian who works in the scientific world of stem cells. So I do laboratory-based research on stem cells. I head up in Cambridge a transplant trial using human fetal cells in patients with Huntington's disease. So I sit on this area. I have scientific medical research which impinges on what I do and why I practice as a doctor, if you like, is actually to try and develop better therapies for patients. I also am a practicing neurologist, so I see large numbers of people who actually have incurable, disabling neurological disorders for which I currently can offer them no curative treatment, only symptomatic treatment. So I would say that there is a tension here. Am I being faithful to my faith in terms of my behaviour with respect to my scientific research and my duties to the doctor? Which of these is the one which I am most answerable to? Well, I would say that this is the most important relationship that I have, and it's the most important one to honour, but there are tensions being pulled upon that from the scientific medical research as well as that from the patient and the demands which they put upon you and your desire as a doctor to make them better. So in the case of Parkinson's disease, I clearly have patients with Parkinson's disease who I wish to be able to offer the best treatment and would ultimately like to be able to cure. I also, as I say, work with stem cells and the medical research which that is developing and the fact that those will impact in the clinic. So I think this is a very difficult position because you have to try and balance this desire to be involved with the development of new therapies which you believe are going to be curative and transform the field. You have that on one hand, you have on the other hand this desire to help patients with conditions for which you can't offer curative therapies, but you wish to be true to your faith and remain true to Scripture. And this takes me on to this issue about where do doctors and where do we sit relative to God and creativity, the sustainer of life and the taker away of life. So in this simplistic schematic, we have God as creator, the conception, and you'll see why I have these separate categories in a minute, from the conception, the fertilization of the egg, to the formation of the embryo, the fetus and the adult. And it's God that creates that, it's God that sustains that, and it's God that takes that away. And in Genesis it quite clearly is stated about man's position with respect to the animal and plant kingdom is that, as is written here in Genesis 1:28, God says, man will rule over every fish of the sea and birds of the air and over every living creature that moves on the ground. So God has given us an authority to rule over living creatures, but the question is, what right do we have as a medical profession with respect to ruling over the life of man? And I think this is an extremely difficult area because, as medical science has shown over the last 50 years, we have an amazing capacity as a medical profession, as a society, to interfere with with the process of creation and sustaining of life. So that I'm not going to talk about euthanasia. Clearly this is an issue in its own right, but the issue of whether one should terminate lives at such time as the patient would like that. But I'm going to be concentrating on these three issues. As a medical profession, we have clearly the capacity to end life in utero through abortion. And that is done through two ways. One is through the medical termination of pregnancy, which is increasingly used, and the surgical termination of pregnancy. So the medical profession have the right or have the abilities to perform abortions, and they are done in increasing numbers, and I'll show you the numbers in a minute. The medical profession also has the capacity to alter has the capacity to administer contraception, many of which is just as it says, it is contraception. Actually, some devices or some ways of performing contraception are actually to allow fertilized eggs not to implant, so one could view that some forms of contraception are actually very, very early stages of abortion. And we also, in the last 30 years, have had the capacity to fertilize eggs outside of the body and to grow the cells in the laboratory and then transplant them back in programs of in vitro fertilization. St. Louise and Miss Cambridge is, of course, very well known for its role in that. So man, the medical profession has the capacity to interfere with this process. The scientific community benefits from the medical profession interference through the use of stem cells and fetal cell transplants, as I've already mentioned. And to give you some idea about how big an issue this is, nearly 40 years ago, 1 in 50 pregnancies ended with a termination of pregnancy. In 2001, 1 in 5 pregnancies ended up with an abortion. So these are vast numbers of cases which are coming to abortion. IVF has clearly also expanded in the last 10 to 15 years. So the medical profession has a very key role in its ability to interfere with this process. And I guess one of the challenges is to know whether this is a right that God has given to man to rule over these aspects, and if we have, what are our responsibilities in terms of how much do we get involved with this, and how much should we be involved with the benefits which are derived from those processes. Now, it therefore begs the question, as a Christian, where do you sit in this network? Now, the easiest way, and I have to say that when I did my PhD 15 years ago, I had to go and give a talk to the Christian Medical Fellowship, and I always remember it because I was going off to give the talk, and someone in the lab said, "Where are you going?" And I said, "Well, I'm going off to talk to the Christian Medical Fellowship about the ethical problems of stem cells." And they said, "Well, there aren't many, are there?" And I thought, well, that seems to me that that's a very good reason why I should go and give this talk. And I felt that God had called me very much to work in this area, and I felt in many ways it'd been very nice if God had not called me to this area, because if I did not get involved with any of this, I would not have to think so much about where, as a Christian, you sit in this whole process. But nevertheless, I am, and I think one has to resolve some of the critical issues which these problems throw up. And I suspect that this is going to be a problem that's going to be increased for those in the medical profession as our technologies increase and our ability to be able to intervene in the creation and sustaining process that God gives becomes more prevalent. So what should our position be? Well, the simplest position, I would say, and what seems in many ways most consistent, is to say, well, as a Christian, I do not wish to be involved in any aspect to do with this. So that I will not be involved with IVF programs. I will not be involved with contraception, although I think there is a distinction to be made between contraception, which is truly that, and early abortion, and I won't be involved with abortion. So that my position is that I will stay out of that field, and that's fine. And as a scientist, I will also stay out of this field because they're a direct consequence of those actions above. Now, that is one position you could take, and that is very much the view that the Catholic Church and some of our friends, particularly a very good friend of mine who works in this field, has made that view quite clear, that as a Catholic he will not be involved in anything to do with stem cells derived from any of these processes, because all of those processes are sinful and therefore we should not be associated with them. I found this quite interesting in terms of IVF, because one of the problems with IVF, as many of you will know, is that the in vitro fertilisation program, which is there to give couples who are having difficulty having children, gives them the capacity to have children through this process. As part of that process, spare embryos are always made as part of that, so that there are spare embryos which are kept as part of that process. And the position of those embryos in terms of their soul, which I'll come on to discuss in a moment, and what you actually do with those embryos, I think is a very important topic, and something that I hadn't really, I suppose, engaged with myself until I was preparing for this talk. So IVF and the generation of spare embryos will generate spare embryos which will be destroyed. You could, however, say, well, actually my position is going to be slightly different. I'm not going to take that view that that all of these acts are sinful and I shouldn't be involved with it. I'm going to accept the fact that contraception has this distinction which I've already made. Abortion is clearly sinful. It is the killing of a life, and therefore I don't wish to be involved with that. But I'm quite happy to work on embryonic stem cells with the IVF program, because I don't necessarily have to destroy the embryo, which is the problem which has been with the using human ES cells. If you use human ES cells, then you need to kill the embryo. And there was a paper 2 months ago which said that you didn't need to do this. So what they did here was they took a series of embryos which had been which are part of an IVF program, been donated for medical research, and in this you have 8 cells. You can take one of those cells, so you leave the other 7. You can test it, which is what they do, to test to see whether the embryo carries a genetic disorder if you're screening for that. But the important point of this was you could take one of these and you could grow up these embryonic stem cell lines. So you can take an embryo, you can grow an embryonic stem cell line, but you don't need to kill the embryo. So that the anxieties about the fact that you are actually killing life, you are killing embryos in this process of making embryonic stem cell lines, was circumvented. And this was hailed as a great advance to get around many of the ethical problems which I've already touched upon and will continue to talk about. However, this paper is not all that it seems, because of the 16 embryos that were used, only 2 cell lines existed. 91 cells were used, which isn't far off the number that were actually in all of these embryos, and no embryos actually survived this procedure. So I think it's important when you read these papers, because this got a lot of publicity. This was in a lot of the papers as a major breakthrough in embryonic stem cell research, which it may be. But actually, when you look at the fine print of the paper, you realise that whilst it does have the potential, the reality is that this technique, as it currently stands, does not get around any of the problems which exist with the use of embryonic stem cells. So I think there are still problems with embryonic stem cells and IVF programs and where one sits with respect to that. So if we now just concentrate on using fetal tissue from abortions, I think one has to ask a number of questions about where does one stand legally and where does one stand theologically. Well, legally, there has been a massive sea change, as many of you will know, in the use of tissue from abortions. If one goes back to the biblical origin, the biblical basis, then a great deal has been made of this passage from Exodus as to whether a distinction is being made between the value of an adult human life, namely the woman, versus that of the fetus. It would imply, or it can be read into this passage, that actually the fetus is regarded as less worthy, if you like, than the mother, and therefore that there's an ambiguity about the status of the fetus, which clearly means that if you are to use fetal material, perhaps that is not as sinful as one would initially believe. I think that this is a misinterpretation of the scripture, and I'll come on to give you some other scriptural texts which I think clearly shows that the creation of the soul, the creation of the conception is actually the beginning of life, and that anything that happens to it thereafter is sinful. It's not to say that these are not issues that we don't have to engage with. There are problems, of course, when a pregnant woman has a complication of pregnancy in which the termination of that pregnancy will need to be considered. And the weighing up of whether the mother's life is greater than the value of the fetal life will have to be taken into consideration. So whilst one can have very clear views, there are clearly difficult decisions that have to be made at certain points in rare circumstances. But if we go back to the sort of legal position, then throughout the 19th century and the early part of the 20th century, there was no ambiguity about where we should stand with respect to the use of fetal tissue and all the derivatives thereafter, because it was outlawed. And the law has changed in terms of its severity for being involved with abortion, but it was quite clear that actually the use of abortion was illegal, and therefore anybody involved with it and using tissue from it would therefore be held as performing illegal acts, whereas of course that all changed 39 years ago with the Abortion Act, which made termination of pregnancies legal under certain circumstances, and the big certain circumstance which creates the greatest controversy is whether a termination of pregnancy should be done for the social and mental well-being of the mother, and that can obviously be interpreted in many different ways. But as a result of this, it is of course entirely legal to have abortions and to use tissue there after derived. It is also entirely legal and licensed that you can use embryonic stem cells or cells derived from IVF programs along with human fetal tissue. It is regulated. There is an authority you have to refer to if you wish to use this tissue. This is not an unregulated procedure. So if I want to work on embryonic stem cells, I will have to get approval from the Human Embryology and Fertilisation Agency that I'm allowed to do that. So it is a regulated process, and Britain very much prides itself on the fact that it has one of the most advanced legislative and informed, it would say, discussions on this, and allows this work to take place. So from a legal point of view, there is no ambiguity, if you like. It's quite clear that you can work with embryonic stem cells and fetal cells. The question is, where do you stand theologically? And I think, to me, this relates to the question on when does the embryo acquire a soul? And I have to say, this is a question which I hadn't really engaged with fully. And once you start to read about this, you realise that throughout the last 2,000 to 3,000 years, this has perplexed a lot of people, such that if you actually plot when people think that the embryo requires a soul, it varies enormously from individual to individual. And what's also quite interesting is that the Greeks clearly distinguished that the soul in the male came much sooner than it did in the female. This was, and it's also quite clear that Aristotle actually had a major influence on this field for almost, well, really up almost to the sort of 400 or 500 years ago, and still has some proponents. In fact, Thomas Aquinas would say that whilst this is the time at which the embryo acquires the soul, he has various stages of acquisition of the soul. So you have vegetative soul, and you eventually end up with the intellectual soul, and that there are various different stages of this. Why is this important? It's important because when the embryo acquires a soul, in my mind would say is the time at which it becomes a person. Prior to that, it is simply a collection of cells. So if you believe that the embryo acquires a soul at 40 days, then why not use the cells before that? They're just a collection of cells. There's nothing special, particularly, about those cells. So many different people have come up with different ages. There is this issue of twinning, which is that up to 14 days post-fertilisation, the egg is capable, or the fetus, or the conception is capable of dividing. So if the soul comes in at the point at which the egg is fertilised, which is my perspective on this, and this is what Calvin said, then what happens at 14 days if that then divides into two twins? Who has the soul? Does one have the soul? Do they split the soul? And this has vexed people, whereas it seemed to me that once you've created a new person, if you like, as it divides, they have a new soul. Some people, and I'm not a philosopher, as you've already guessed, would say Locke, for example, has this view on rationalism, that actually soul is when you can actually rationalise things, which would therefore, if you take it to its logical conclusion, would support infanticide and things of that nature. So this is very important, but it would seem to me that the scriptural basis is for the soul being there at the moment that the egg is fertilised. And if we look at three passages, this is a very famous passage from Psalm 139, which I think quite clearly lays out how God is there at the moment in which that egg is fertilised and that person comes into being. There are also three other quotes here from Jeremiah, Galatians and Ecclesiastes, which again, I think, clearly give a scriptural basis to the fact that the soul comes about at the time at which the fetus is, the fetus comes into existence, namely that the egg is fertilised. So I would have a position there. That then raises a question, well, actually, if you believe that the soul comes into existence at that point, then all of those acts are sinful. They are the taking away of a life. So whether it be collecting ES cells from an IVF program, whether it be collecting tissue from fetuses at any age, all of that is a sinful act. So then one is left saying, well, actually, if it is a sinful act, is there any time at which one can engage and be involved with that process? So you can have the pragmatic approach, which is where I'm going to conclude and say where I stand, which many people would regard as a very weak position, which is that I do not condone abortion. I think abortion is sinful. I think it is wrong in the majority of cases, although I think to be black and white about this is wrong. I think there are circumstances in which one could justify it. However, abortion is going to happen. There are 200 or 1,000 abortions a year, so this will happen. So isn't it better to actually do something useful with the tissue which will come about through abortion? And there are very clear guidelines on this, so that the Department of Health and the Polkinghorne report have very clear guidelines that those who are involved with this aspect of work, which is where I sit, have nothing whatsoever to do with this. So that the mother who comes up for a termination of pregnancy agrees to a termination of pregnancy. Once she has agreed to that, she then is asked whether she would like to donate that tissue to medical research. If she agrees to that, then we are alerted to that fact, and when the termination happens, we then collect that tissue and we then use it. But we are in no part involved with the consenting procedure, the procedure itself, or any decisions which are made on that process. So there are very clear guidelines which I think are important to emphasise. So the pragmatic approach is abortion will happen, why don't I just use the tissue? But then becomes the issue of whether you are actually, by so doing, indirectly condoning abortion. You are actually encouraging it, because now there can be an argument that actually I'm not sure whether I want an abortion, but I'll have an abortion because it's going to go to some good purpose, and I might be able to help some people with incurable diseases. But I think this then raises questions as to the degree of cooperation that should exist between the person generating the tissue, if you like, those using it for research, and the possible cures that might arise out of it. And if you take a view now that you're not going to get involved with it because you are in part cooperating in this procedure, at what point do you then say in the future, I will have nothing to do with it? So if an ES cell becomes a cure for Parkinson's disease, and you are a neurologist in a clinic, and people say, I want that cure for Parkinson's disease, do you refer them to someone else? Do you have the right to say, I don't believe that that's the correct procedure, and I therefore feel that you should not have it? So where do I see myself sitting with this? This, again, as I say, is a very personal view. As a Christian, I think life is sacred, and life is sacred from the moment in which it's created, from which that egg is fertilised. As a pragmatist, abortion will happen, so it is going to happen, and that tissue will be generated. As a neurologist, I will see lots of people with neurological diseases which I cannot help. This will become an increasing problem as the population ages. As a scientist, stem cells have a real potential. I think there's a real scientific argument to be made for their use and their ability to cure people with neurological diseases. And I think if you don't get involved as Christians in this type of work, then I don't think it will be properly discussed. It will not be discussed by people who actually understand the reality of stem cells. It will be discussed by people who have other agendas, people who don't have a Christian perspective on it. And as I say, I presented my stance on this. This is why I do what I do, and these are the reasons why I do it. I don't know whether this is a terribly strong position, whether this is a terribly weak, half-hearted sort of position that I put myself in. I don't know whether it's the right thing, the wrong thing. And it is a constant prayer request of mine as to whether this is something that I should be working in this field, or whether this is something that I have misinterpreted what God has sent me to do, and I've therefore been involved with an area of work which it would be better for me not to be involved with. And with that, I will see.
Thanks very much, Roger. We've got a little bit of time here for questions. I'm sure this has provoked plenty of questions. There's one in the back there, yes.
I think it to be taken as read that anyone who thinks there are no ethical problems in abortion hasn't actually performed them. And I'm glad to hear you say quite unequivocally that you believe that's wrong. IVF is also wrong for a good many reasons, but one of which is, as you pointed out, that you make so-called spare embryo who's going to be destroyed or experimented on. You know you're going to do that, and therefore that's wrong. My question is that if you could develop satisfactory stem cell therapy for Parkinson's disease, doesn't mean cells, you go to your patients, you want to help them, of course you do, you're a doctor, and you advise him to have this operation to implant these cells, which you know in your very active conscience, it's come through how active it is, you know it's wrong. You know this therapy shouldn't be given, shouldn't be used. Are you doing your best for that man? You are trying to do your best for him as your patient, but he has a soul too. Now this, you asked a question as to whether or not, if it becomes routine therapy, established practice, not the exceptional practice things that happen by drift really rather than by formal and open debate, so I'm glad you're encouraging. If comes a time when it is a standard practice and a patient is referred to you, and you feel that this shouldn't be a therapeutic option, it's not in your therapeutic armamentarium, then I think you should say, awfully sorry, or whatever term you use, but you'll have to go and see somebody else, because I think it's wrong, and I'm not going to get involved with that anymore. I did, perhaps, but I'm not going to do it now. And I say that from a position of experience, because heart transplant started in Papworth in 1980. And I, by that time, found out what it involved. It involves taking beating hearts out of people who are not really dead, that are only notionally dead. This invention of this brain death nonsense, which is now, fortunately, falling in this house. But this was going on. I wasn't prepared to be part of that, because I thought that taking a beating heart out of someone who wasn't dead was murder. And I thought, therefore, this procedure was wrong. So I said, and when they were referred to me subsequently, sorry, I'm not going to be involved in that. You go and see someone else who may be. I got into a lot of trouble over that attitude. As you know, I eventually had to resign my rather satisfactory job on those grounds. But I've slept well since.
No, and I think that encapsulates the nature of the problem. I think I think the trouble is it's very difficult sometimes to divorce this. If you have patients who you know extremely well, that you followed for many years, you develop a very close relationship with those patients, and if you think you can offer them something which could cure their condition, it's quite hard not to be able to offer them that. Now I think it's entirely right to say, well, from my perspective I can't support that, but I will refer you to somebody else who will offer you that treatment. The question I would then have, well, have you just carried on? I mean, have you really changed your position? Because in many ways you're still supporting that therapy, because now you've asked somebody else to give it to them. And in that case you're sort of saying, well, I think the therapy is worthwhile having, I'm just not prepared to do it myself. So I think they're very difficult issues, but I think what you say is absolutely sort of nub of the problem, those tensions which you have.
Questions from this side? Wait, yes.
One thing you didn't mention explicitly, but it might be because of the practical problems, was umbilical stem cells. What are the ethical and practical issues involved in umbilical cells?
Well, I think that I would put those, if you like, more into the sort of adult stem cells, if you like. That I think they do have they clearly seem to have a source of which most people would not have any great issues. I think the question is whether you can truly do with them what people say you can do with them. And I think the news item today seems to imply that you can do a lot with them. I think a lot of claims that you can turn them into nerve cells are not true. And I certainly have a lot of discussions but a number of discussions with patients who ask that one of their families having a baby, should they bank the umbilical cord cells? My answer is no, because there's no evidence that they will actually do what people claim they're going to do.
Okay, so in your opinion, you lump all the adult stem cells together because you think they have the same potential together, whether they be blood ones, marrow ones, umbilical.
I think there are a lot of common problems with all of those adult stem cells.
Yes.
Would it be possible to use animal sources of stem cells? Is that too difficult?
Can you repeat the question?
Yeah, it was whether you could use animal stem cells, which is a very good question, which I've spent quite a bit of time working on, is whether one can use animal stem cells. The problem with animal stem cells is, in a nutshell, if you use animals, your body will react very strongly against them. And so the rejection is a really big issue. The reason why you can't do pig transplants in this country, or anywhere in Europe at the moment, is not to do with that, the rejection issue, but is the anxiety that the foreign animal tissue will put a new infection into man. So the argument is that HIV may have come about through monkeys in Africa, and therefore it jumped, and new variant CJD is an example of where you can jump across species. So if the monkey gave us HIV, then if we put pig cells, for example, into a patient, could they develop some new disease? And therein lies a whole series of problems, because if the patient then develops a new disease, who else is at risk of having that new disease? Should that patient then be allowed to carry on living in the community because they may have an infection which will spread to others? Should they all be isolated? Who's responsible? Are the government responsible? And so on. And so I think the use of pig tissue in this country almost certainly will never happen.
Okay, this is a really huge discussion. I'm sure we could easily go on for an hour, and I'm sorry, but the time is actually up, because we do promise everyone we'll finish spot on 2, which is just about a few seconds. So I think we'd really like to thank Roger very much for a very informative but very honest talk as well. I think we'll to